get to do it. Okay, and we will probably, I'm thinking we'll start with chapter 13, which is what we didn't get to last time. And then if you guys had any questions on anything, we can cover it. But otherwise, we'll probably just loop back around to chapter 4.
Okay.
Cool. So for chapter 13, first we've got our terms. And the first 3 terms are all kind of like similar to each other in a way. So the differences are kind of a little bit subtle. So what's systematics?
The study of diversity of life with focus on relationships.
Yeah, exactly. And what's phylogeny?
I think like how the relationships came about, like the evolution.
Yeah, exactly. So phylogeny is the study of the evolutionary relationships between organisms. And then in systematics, you're studying the overall diversity of life with a focus on the evolutionary relationships. So phylogeny is a big part of systematics, but it's not the only part, because in systematics, you're also looking at things like morphology or the characteristics of organisms, not just how they're related. And then what's taxonomy?
Taxonomy. The classification of organisms?
Yeah, how you're actually classifying organisms. And that would be guided, of course, by phylogeny and systematics. So you're kind of grouping organisms, ideally, in groups that are, containing species that are closely related to each other. And what's a phylogenetic tree?
It's a tree that like a tree that shows like like how everything's related.
Yeah. So yeah, exactly. That's a tree that's similar to a family tree, but it's showing the evolutionary relationships of like how recently 2 species diverged from each other, or how recently 2 groups of species diverged from each other. Then what's an allele?
A version of a gene.
Yeah. A specific version of a gene. So if you have a single gene, you might have multiple alleles for that gene. Each allele would make a protein that's a little bit different, but is still the same basic protein. And then what are homologous genes?
Genes from the same ancestor?
Yeah, exactly. Genes that have come from the same ancestor. So that would mean that the ancestor was some species that these 2 modern species had both evolved from, and that ancient species had this gene. Now these 2 modern species have 2 versions of the gene that are a little bit different because, of course, there's been evolution and mutations occurring. But since the genes are coming from the same ancestor, they are homologous, or they're coming from the same place. Then what's an ortholog?
They're homologous, but they have like the same function.
Yeah, exactly. So homologous genes that have kept the same function are going to be orthologs. And then what's a paralog?
It has a different function.
Right. So those are homologous genes where at least one of those genes has evolved a new function, but you can still tell that it's related to its homolog, or to its other paralog, I should say. And what's a type strain?
A strain based on which a microbial species was first described.
Right. So for any microbial species, it's kind of hard to define it. You're going to have multiple strains that belong to that species, but the very first one that was discovered and characterized that the species is based on, that's the type strain. What is the RNA World Hypothesis?
I'm just gonna read it off the top. Is it the idea that the RNA precedes DNA and protein and was used to store information and do work by first organisms?
Yeah, yeah, exactly. So, right, the RNA World Hypothesis is stating that RNA evolved before DNA or proteins did, and it was basically serving the functions of both of those molecules. So today, DNA is used for storing information, protein is used for doing work. So in the early RNA world, before you had protein or DNA, RNA would have been storing information and doing work at the same time. And your earliest organisms would have had RNA as kind of their main molecule, doing all the work of the cell and storing all of the cell's information. Then later on, you would have evolved DNA and proteins. Cool. And what's endosymbiosis?
Mitochondria and chloroplasts were once independent bacterial cells.
Right. Right. So that's the idea that these organelles, mitochondria, and the chloroplasts came from bacterial cells that were kind of engulfed by a larger eukaryotic cell, and instead of dying, they just lived there happily and kept dividing and eventually became organelles, the mitochondrion and the chloroplast. What's some of the evidence that supports that theory?
Do they have two membranes?
They have two membranes, which
Different DNA.
Their own DNA.
right, they have their own DNA, they have their own ribosomes. What did you say, Noah?
They,
It has different DNA.
Right, the DNA in your mitochondria and chloroplast is its own DNA, but it's also different from the DNA in the nucleus. It looks more like bacterial DNA than like eukaryotic DNA.
they divide independently.
Right, and they divide independently. So all the other organelles are dividing whenever the whole cell divides along with the nucleus. Your mitochondria and chloroplasts with their own DNA and their own ribosomes make their own proteins, and they divide according to their own schedule. Then what about when you look at the mitochondrial and chloroplast DNA? Who can you tell that they're related to?
Your mom's side?
Sorry?
Isn't it? Don't you get it from your mother?
Right. Oh, well, yeah, actually, if you're looking at a person's mitochondrial DNA, then that's coming from the mother. So you can kind of look at the mother's or the maternal line ancestry by checking out someone's mitochondrial DNA.
Bacteria.
Yeah, bacteria. So for mitochondrial DNA, if you're looking at like the nitty-gritty details of it, you can tell something about a person's mother and the maternal line that they're descended from. But when you're looking at the more kind of broad strokes, you can see that it's like bacterial DNA, and it's like specifically it's like the DNA for specific bacteria. So for mitochondrial DNA, it looks like proteobacteria DNA. For chloroplast DNA, it looks like cyanobacteria DNA. Cool. Okay, then the Great Oxidation Event. What is the Great Oxidation Event?
Oxidation of Earth's atmosphere in response to rising oxygen.
Right. Where is that oxygen coming from?
Plants?
Not yet. This is going to be before plants evolved.
Would it be like fixation? Like
it is related to carbon fixation.
Is it from the bacteria cyanobacteria? We learned it like in the other chapter.
Yeah, exactly. Yeah, it's cyanobacteria. So the cyanobacteria evolved oxygenic photosynthesis, the first type of photosynthesis that creates oxygen, which is the same type that plants use, and they actually got it from cyanobacteria. Once these cyanobacteria started manufacturing oxygen, that's the first time that you had a lot of oxygen in the Earth's atmosphere. And so that caused oxidation of a lot of things, since oxygen is very good at taking electrons. So this oxygen is getting reduced. All the things around it are getting oxidized by it. What are some of the consequences of the Great Oxidation Event?
The anaerobes, they died.
Right. Anaerobic bacteria or archaea that couldn't handle all that oxidation, they died. And they were only left living in places where no oxygen was around, like deep in the underground or underwater. What's another consequence?
The aerobic respiration evolved.
Right. It lets you do aerobic respiration. It was impossible to have aerobic respiration, which needs oxygen for an electron acceptor, before you had a lot of oxygen in the atmosphere. So because of the Great Oxidation Event, we got aerobic respiration, which is the type that you and I use. And you know, we need it, so it's good. And then one more consequence.
The ocean changed the color?
Yeah, ozone layer. That oxygen is able to react to create ozone in the upper atmosphere, which then protects us from UV rays. Cool. What is horizontal gene transfer?
Gene exchange apart from heredity?
Right. So gene exchange apart from heredity, meaning that normally we think about genes being exchanged in heredity, genes going from a parent to the offspring. In horizontal gene transfer, something else is happening. So there's multiple ways that it could happen, and you don't need to know the details of those different ways. Just something else is happening to transfer DNA that's not from a parent to its offspring. Actually, endosymbiosis is an example of that. So you have, cyanobacteria DNA in chloroplasts in plants, and they didn't get it by being descended from cyanobacteria. They got it through endosymbiosis, which would be a type of horizontal gene transfer, because it's exchanging genes, but not through heredity, not through parents to offspring. So why does horizontal gene transfer make it hard to study phylogeny and determine evolutionary relationships?
Wait, can you repeat the question? I'm sorry.
Why does horizontal gene transfer make it hard to do phylogeny and study or figure out evolutionary relationships between organisms?
Would it be hard, like, to determine where the genes came from?
Yeah, exactly. So you'd like to be able to assume that if two organisms have the same genes, they got it from the same place, from a common ancestor, and those are homologous genes that they both got from a common ancestor. So that's kind of the assumption you need to make to do phylogeny, because that's really the evidence that we have about all these ancient organisms. It's just DNA that's been left behind. But in horizontal gene transfer, you can have organisms getting genes that don't have a common ancestor with whoever has that homologous gene. So like plants and cyanobacteria, they don't share a recent common ancestor. You would have to go back basically to Luca, or I guess to where Archaea diverged from bacteria in order to find their common ancestor. So if you were just going off the fact that they have cyanobacterial DNA in the plant chloroplasts, you might assume that they're more recently related to each other than they actually are. So that's not really a big problem for multi-celled life forms, for eukaryotes, because they don't really do horizontal gene transfer very much. There is some, but not a lot. But for prokaryotes, it's more common. It makes it really hard to figure out how microbes are related to each other.
What would it be like rare for it to happen, or does it happen like a lot?
It is rare for it to happen, and it has happened a lot.
Okay.
Because there's just so many microbes out there and so much time that has passed. even for eukaryotes, like multicelled creatures like us, we can get some horizontal gene transfer that comes from viruses. So there's some viruses that will inject their DNA into your cells, and it'll kind of incorporate into your nucleus and just insert itself into your chromosome. And then it's supposed to get cut back out when the virus is going to make new viral particles. But sometimes that doesn't happen right, and it ends up cutting out a little bit of your DNA as well. And then that new viral particle is going to get that little bit of your DNA, and it's going to go infect some other host and bring them your DNA. And that has actually happened enough times that it's kind of left a permanent mark on our genomes. So it's estimated that maybe 8 to 11% of the DNA in your genome is actually coming from a virus. But in order for that to kind of enter into like the overall genome of the human species, it's really rare because you would have to have the virus infecting a germline cell that's actually going to give rise to a sperm or an egg cell that would form, offspring. If this is a virus just infecting like your skin, that new DNA is just going to stay there with you. It's not going to go to your offspring, so it won't enter kind of the overall genome of the species. So it's really rare for that to happen, but over the full scale of time, it has also happened quite a lot. If like about 10% of our genome isn't even human. It's actually really interesting.
Yeah.
Okay, so next we have evolution. So what is evolution?
It's a change in, I guess, different animals or species over time, or population over time.
Yeah, exactly. A change in the characteristics of a population over time. Not for an individual animal, but for a population or a whole species. So you could look at that in terms of the characteristics of that population, like the things that you can see, but of course that's all going to be based on their genetics, or at least, environment also plays a role, so a large part of it is going to be based on genetics. So you could also see evolution as a change in allele frequency in a population over time, or change in, which alleles are present in that population and how many individuals would have that specific allele. So actually for the test, I think I've got both definitions on there. Let me just double check real quick. Yeah, both definitions are there. So if you want to remember characteristics changing over time or allele frequency changing over time, either one is good. What are the sources of genetic diversity for evolution?
Like mutation?
Mutation, exactly. That's a big one. And then there's one more.
Recombination?
Recombination, yeah. So mutation is where you would be getting most of your new alleles from. It's where you have an actual change in the genetic sequence that's then passed on to offspring. Recombination is more like mixing up what you already have. So you could have, if you're an organism that has multiple chromosomes, you can have different pieces of different chromosomes kind of exchanging with each other, and genes would just be moving around. But that can also create new alleles because it's not like genes will always be transferred as a single unit. You might have like half a gene get transferred to a new spot in a new chromosome, not necessarily a whole gene. And so that mix-up can kind of make new alleles, even potentially new genes. So those are your two sources of genetic diversity, which you need to have evolution occur. Then you also need some process to drive evolution. What are the two main processes that drive evolution?
Natural selection and genetic drift.
Yeah, exactly. How does natural selection work?
If you don't change to your environment, it'll just die out.
Right, that's part of it. So organisms that are not well adapted to their environment are more likely to die, which leaves behind the ones that were well adapted to their environment. So I guess on a more detailed level, in natural selection, everybody has, different traits. Some of them are advantageous for an environment. Some of them are disadvantageous. Some of them are neutral. Whoever has advantageous traits is more likely to have more offspring. And then those advantageous traits will become more common in the population in the next generation. And then if you have disadvantageous traits, you're less likely to have offspring. And then those traits are going to become less common in the population in that next generation. So technically, it actually doesn't really matter if you die. Dying only matters insofar as it affects your ability to have children. So if you lived like a nice long life and you had many children, and then at the end your trait killed you, the evolution doesn't care, or natural selection doesn't care. You are fit. You had many children. And those traits are being passed on in a large proportion to the next generation. And then what's genetic drift?
Is in the allele frequency traits.
What type of changes in allele frequency happen in genetic drift?
Random changes?
Random changes, yeah. Right, so in genetic drift, it's drift because it's just random changes in the allele frequency, which would happen like maybe if you had the best traits in the whole world, but then, you just had an accident, like maybe you got in an unfortunate car crash and you were killed before you could have children. So now the next generation is not going to have all those nice traits that you had. They're gone forever, and it's not because they were disadvantageous. It's just because random things happen. And let's see, what type of population is more likely to be affected by genetic drift?
Like, smaller population?
Smaller populations, exactly. The smaller the population is, the more likely it is that a trait can just vanish because everybody who had it ended up dying due to some random cause. If you have a really big population, it's hard to randomly have everybody with a specific trait die. It can still happen over time, but it's less likely. What are some characteristics of microbial evolution that are, I guess, kind of distinct to microbes?
They adapt fast?
Right, they adapt very quickly. So when the environment changes, very quickly you're going to see that they're gaining new traits to meet the challenges of that new environment. What else?
They evolve quickly.
They evolve quickly, right? So that's related to the first one where, they're able to change their traits very quickly in response to the changing environment. Why can they change their traits so quickly and evolve so quickly?
They're smaller in size?
They're smaller in size, that's part of it. So they grow real fast. So their generation time is real fast. Like for humans, it's like, well, I don't know, it depends. 15, 20 years. It's a long time. But for microbes, it would be like some hours or maybe some days. So they're going through generations basically like lightning. And with every new generation, you have that opportunity for new mutations, new recombination events, for more advantageous traits to arise and become common in the population. Also, for some microbes, for prokaryotes specifically, the fact that they're haploid, so they only have one copy of every gene that they have. Unlike you have two copies of every gene. You're diploid. So one copy can kind of mask another copy in diploid organisms, but in haploid organisms, since they only have the one copy, whichever copy they got, that's what their trait is going to be. What's another characteristic of microbial evolution?
Speculation?
The different strains have different genomes?
Right. So if you're looking at a microbial species, all the different strains in there are going to have significantly different genomes from each other generally. They're not going to be like all looking the same at a genetic level. So then you would have kind of two parts of the genome, the part that all of them have, and then the part that only each individual strain has. What's another characteristic of their evolution?
Speculation?
Sorry?
Would it be like they speculate slowly?
Oh yeah, they speciate slowly. So they evolve quickly, they gain new traits quickly, they lose bad traits quickly, but when it comes to a strain actually forming a new species, that happens real slow, which is probably good, or else it would be just ridiculous to categorize them all and do the taxonomy. Right, so but that's partly why you're going to have so many different strains for a species. All those strains are living in different places. They're going to have a little bit different environments. They're going to end up with different genes as they adapt really quickly to their environments. But they're all going to stay remaining like part of the same species for a long time. So they speciate very slowly. What's one more characteristic?
The non-beneficial genes are deleted from the genomes?
Right. So if there is a gene that is not useful for a microbe in its current environment, it's probably going to be deleted soon from the total population's genome. So you're going to have like one microbe that ended up having that gene deleted as part of a mutation or recombination event, and that microbe is actually going to be a little bit better at living in that environment since it's not making something that isn't useful. So it's going to end up dividing just a little bit more quickly when it's not wasting that tiny bit of energy making something that wasn't useful. And since the generation time is so fast, very quickly, you're going to see that that microbe has taken over, or I should say its progeny, its offspring, have taken over the population. And now nobody has that gene that wasn't useful anymore. All right, so microbes are going to have gene deletion occurring very quickly, and that's part of the reason why their genomes are really small on average. Okay, and then finally, why is it hard to define what is a microbial species?
Is it because it's based on like sexual reproduction, which most microbes don't do?
Yeah, exactly. Right, so the normal definition of a species is that it's, you know, a group of individuals that are able to interbreed with each other and produce viable offspring, which would be offspring that can then have children of their own. So that's based on interbreeding. It's based on sexual reproduction. So that species or that definition of a species can't work for any organism that is reproducing asexually, which all prokaryotes reproduce asexually. Most microbes reproduce exclusively asexually. A few of the eukaryotes that are microbes are able to do both asexual and sexual reproduction. But overall, microbes are not reproducing sexually. So you cannot use that definition for them. It just doesn't apply to them at all. So what is the definition that is kind of the most accepted for a microbial species?
A group of strains with similar genetic characteristics that share a recent common ancestor.
Yeah, exactly. So we're kind of defining the species based on its evolution and based on the characteristics that it has. So if you have two strains that have that recent common ancestor and you see that their characteristics are pretty similar, then you're going to say that they're part of the same species. If you see that they have similar characteristics, but their most recent ancestor is like a long time ago, then you're probably going to say that they're different species. But it's kind of nebulous So like basically it's not well defined, and to some extent it's actually a matter of opinion. What is a microbial species, or are two strains belonging to the same species? It's a mess. Okay, and that's chapter 13. So is there anything in the study guide that you guys like wanted to go over?
For me, it was like chapter 4 was like the longest, and yeah, I just kind of need a little help with that.
Yeah, for sure. Did you have anything, Noah?
Looking over the study guide, no, I don't think I had any other questions.
For sure. Okay, cool. So yeah, we will loop back to chapter 4. Okay. I guess I'll just start at the beginning here, or should I skip the beginning part of it?
The terms?
Yeah.
No, I'm good with the terms. I think like from the basic structure all the way.
Okay,
like,
for sure.
yeah.
Cool. Okay, so what are the 3 parts of every nucleotide?
3 parts.
The sugar, the base, and the phosphate?
Yeah, the sugar, the nitrogenous base, and the phosphate group. So that's the main part of like the basic structure of a nucleotide, that you got your sugar and a phosphate group attached and a nitrogenous base attached. And then you have the carbons in that sugar numbered 1' through 5'. So what would you find attached to the 1' carbon?
The base?
The base, yeah. Then what's attached to the 5' carbon?
The phosphate?
The phosphate group, exactly. And what's going to happen at the 3' carbon?
It's going to have like a phosphate bond, but like it's going to connect the other nucleotide with it.
Right. That's where you're going to make your phosphodiester bond to join that nucleotide to another nucleotide. So that means that the phosphate group of your new nucleotide is going to attach to that 3' carbon on your existing nucleotide.
So,
and it has to go that way. It can't go the other way around.
so for
general.
every nucleotide, if it's connecting to another nucleotide, it's going to always be on the 3'. It'll never be on a different prime.
Right. Well, I shouldn't say never. There are a few examples of limited times when it does happen. So for instance, the 5' cap, that's actually the 5' phosphate attached to the 5' phosphate on another nucleotide. But that's really, that's the only example that I know of, to be honest. So your regular polymerases that are building strands of DNA or RNA, they can only add to the 3' carbon. They can't add anywhere else.
And then so that from that 3' carbon, it's going to be attached to the other 3' carbon, or will it be attached to a different carbon?
So you're going with your 3' carbon on the first nucleotide, and then the phosphate group is going to come in on your second nucleotide and attach. And then that phosphate group, of course, is attached to the 5' carbon on its own nucleotide.
Oh, okay. Then yeah. And then it goes 3, and then to phosphate, and then 5.
Yeah, exactly.
Okay.
Cool. So what are the different nucleotides that you can have? What are the 5 nucleotides you can have?
A, T, C, G, and U?
A, T, C, G, and U, yeah. How are they going to base pair with each other?
So I think A goes with T, and then C goes
Yes.
with G, and then for RNA, A goes with U. And not
Yeah, exactly. Yeah, so in DNA, you have A pairing with T, C pairing with G. In RNA, A pairs with U, and C still pairs with G. What is that base pairing based on? Or what type of interaction between the two bases is creating the base pair?
like the sequence.
Not the sequence. It's a type of bond, actually.
Is it the alpha or the beta one? Those ones.
Let's see, that would be. Well, I guess there's multiple things that could be. um, it's actually going to be a hydrogen bond.
Oh,
So
okay.
your bases are going to have like some oxygens and some nitrogens that are having like hydrogens attached or not, which would be places where you can do hydrogen bonding. So with your C and your G, that pair can make 3 hydrogen bonds. And with your A and your T or your A and your U, that pair can make 2 hydrogen bonds. Then what's the primary structure for nucleic acid?
Well, I'm sorry. So you said, what is it based on?
Yeah, so it's based on a hydrogen bond, or that is to say
Hydrogen.
the interaction between two bases that lets them make a pair is the hydrogen bond. They come together and they're able to form 2 or 3 hydrogen bonds with the other base that's next to them.
Okay.
So then it's those hydrogen bonds that's like holding that base pair together. Cool. And then what's the primary structure for RNA and for DNA?
Would it be the helix for DNA?
For DNA, that is the structure, but that's not the primary structure. That's a different level of structure.
Is it a sequence of base in a strand?
Yeah, the sequence of nucleotides in the strand or the sequence of bases in the strand. I guess we kind of use base like interchangeably with nucleotide. Right, so it would be, that's the actual sequence, so like A, T, C, G, 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 that's the primary structure.
Okay.
Then for DNA, the secondary structure would be what?
DNA.
T.
The double helix?
Yeah, that one's the double helix. So for DNA, in the secondary structure, you get your 2 strands lined up together, they base pair with each other, and then that coils up to form the double helix. And I guess I already answered the second part of that. The secondary structure for DNA is based on base pairing between the 2 strands, so it's based on hydrogen bonding between the bases on each strand. Then what is the secondary structure for RNA?
How the base pairs, strand base pairs with itself?
Yeah, so RNA is usually just one strand, so that strand is going to kind of wrap around different parts of the strand, will be able to base pair with, other parts of the strand, and that forms simple little structures like stems, loops, usually stems and loops. What interaction is that based on? I guess you already kind of said it though. Well, since you already said it, it is base pairing. Right, so the strand is base pairing with itself, so it's that base pairing or those hydrogen bonds between bases that is holding the secondary structure together for RNA and for DNA. Okay, so that's our basic nucleic acid structure and stuff. Then we have a little bit on proteins. So what's the basic structure of an amino acid?
Is that the amino group, the carboxyl and the central alpha carbon?
Yeah, that's part of it. So you have kind of like 2 parts to the amino acid. One of them is the backbone, and then in the backbone you've got your amino group, your central alpha carbon, and your carboxyl group. Then what would you have attached to the backbone?
Side chain?
The side chain, yeah, exactly. So every amino acid will have the same backbone, but a different side chain. And in the backbone, you always have the amino group, the central alpha carbon, and the carboxyl group. What do we call the bond that forms between two amino acids?
Peptide bond?
Yeah, peptide bond, cool. So for nucleic acids, that's a phosphodiester bond. For amino acids or proteins, that's a peptide bond. Then we can group amino acids based on their side chains. What groups would we form? Or how can we group amino acids by their side chain?
Is it charged like polar and the non-polar?
Yeah, charged, polar, and nonpolar. And then in some systems with the charged ones, you could also divide it into acidic or basic, depending on if they have a positive or a negative charge. But we'll just stay with charged, polar, nonpolar. Cool. So what's the primary structure of a protein?
The sequence of amino in the polypeptide chain?
Yeah, the sequence of amino acids in the polypeptide chain. So technically, we don't call that polypeptide a protein unless it's like fully got all its structures, it's folded upright, and it's functional. Then it's a protein. Otherwise, it's a polypeptide. Right, so the sequence of amino acids in that polypeptide or in that protein, that's the primary structure. What's the secondary structure for a protein?
It's the folding of the peptide bonds to form the helix and the beta sheets.
Yeah, exactly. So simple folding of that polypeptide chain to make either an alpha helix or a beta pleated sheet. Then what's the tertiary structure?
The complex folding?
Yeah, complex folding of the polypeptide, which would make unique 3D shapes. And then finally, the quaternary structure.
The arrangement of multiple polypeptide subunits to form the full protein.
Right. So for any protein that has subunits, each one of those subunits is its own polypeptide that would have primary, secondary, and tertiary structure. And then how you're sticking those subunits together is going to form the quaternary structure of the final protein. They have to be arranged in the correct way for it to function correctly. So for all those levels of structure, primary, secondary, tertiary, and quaternary, which is the level that not every protein has?
The fourth one?
Yeah, the fourth one, quaternary structure. So every protein is going to have a primary, secondary, and tertiary structure, but not every protein has subunits. Only proteins that have subunits are going to have a quaternary structure. Cool. So that takes us to replication. So what is replication?
The process of copying chromosomes or plasmids,
Right.
like cell division, basically.
Right. It's copying the DNA. Generally, just copying the chromosome, but if you have plasmids, you would copy those too. And it's always done in preparation for cell division. So it's kind of a complicated process with a lot of different enzymes involved. What does the helicase do?
Separates DNA strand at the origin.
Right. And what's the origin?
Is the origin, would it be at one of the ends?
It's actually going to be, for a circular chromosome, I guess.
Oh, yeah.
I wanted to say it's going to be in the center, but then I realized on a circular chromosome, there's not really a center. In a eukaryotic chromosome that's a line, the origin, you're going to have actually multiple origins that are kind of dotted all along the whole chromosome. Then for a circular chromosome, you will just have one somewhere, and then we could arbitrarily say that that's the center. But it's not going to be like right at the end. If it's a linear chromosome, it won't be right at the end.
So, like, when you ask, like, what's the origin, like, define what the origin is?
Yeah, or what happens at the origin, I guess.
So I'm looking at the model, and so it's showing that like the double helix is unwinding at the origin.
Yeah, so if you guys are probably overthinking it a little bit, but the origin is just a place. It's just a place where replication begins. So that's the place where helicase is able to attach and start replication. So at that origin, you have like a specific nucleotide sequence, and then the helicase enzyme is able to recognize that sequence. So it's going to come in and bind there at the origin, and the origin basically is just allowing it to attach to the DNA. Once the helicase is attached there, then it does its job of separating the two strands of the double helix. Cool. So once the helicase has separated the two strands, what's the next protein that comes in?
Is it the single strand DNA binding?
Yeah, those guys.
Okay.
Single-stranded DNA binding proteins, or the SSBPs. They will come in and do what?
They will bind those separated strands to prevent base pairing.
Right, exactly. They're coming in, following the helicase, binding to those two strands that the helicase has separated, just making sure that they don't come back and base pair with each other again, which would reform the double helix. The double helix needs to remain open while replication is occurring. Then in front of the helicase, you have another enzyme acting. What's the enzyme that goes in front of the helicase?
Primase?
That's going to be coming behind the helicase. That's the next enzyme that would come in behind the helicase, actually.
Oh, so would it be tropase? Tropase?
Yeah, topoisomerase or gyrase, depending on which organism we're talking about. But actually, a gyrase is just a type of topoisomerase. So you'll have this topoisomerase or gyrase enzyme come in. It's coming in ahead of the helicase, which means that it's actually attaching where you still have the double helix intact. What does the topoisomerase do?
It unwinds the DNA?
It unwinds the DNA.
And it'll rejoin.
Yeah. Yeah, by making a cut. So it nicks one of the strands, lets that strand unwind around the other strand, and then heals the cut that it made. What's the purpose of doing that?
Is it, I'm reading, it says it relieves the twisting forces.
Right, yeah. So it's like if you had like a rope and you're going to unwind the strands of the rope, you're making it straight on your end, but on the other end of the rope, it's getting like extra twisty. All of those twists are just bunching up at the other end. So the topoisomerase is basically just removing those extra twists that are introduced when you separate the two strands. And if you didn't do that, it would end up coiled so tightly that helicase would not be able to separate the strands anymore, and then you would be stuck. So that's the topoisomerase. And then now off you mentioned that the primase comes in behind the helicase. So that would be the next enzyme that's involved in replication. What does that guy do?
It synthesizes RNA primers?
It's going to, It's going to synthesize an RNA primer, or a primer made out of RNA. What is the job of the primer?
To start off the daughter strands?
Yeah. It's gonna basically form the starting point for the DNA polymerase to start making its daughter strand of DNA. So why does the DNA polymerase need a primer to get going?
Because it has the two limitations. It cannot start the strands from scratch.
Yeah, exactly. So RNA polymerases can start a strand from scratch. DNA polymerases cannot do that. They need an existing strand that they can add nucleotides to. So primase is actually a type of RNA polymerase that's going to just make that little primer, that little segment of RNA, and then DNA polymerase will have somewhere to kind of grab onto and start adding nucleotides. So once you have your primer, what's the next enzyme that's going to come in?
The ligase?
That's actually going to be the last one.
Oh. Is it the DNA pol 3?
Yeah, DNA polymerase 3 or DNA pol 3 is going to come in next after the primer is done. And what does DNA pol 3 do?
Builds daughter strands?
Yeah, it's the main enzyme that is building the bulk of the daughter strand. Right, so it just attaches to that primer, starts adding nucleotides to the end of the primer, and as helicase is going to be continuing to walk down the DNA, continuing to separate the double helix, and DNA pol 3 is basically going to be following behind it, adding new nucleotides to the daughter strand based on one of the original parent strands. So it'll keep doing that until, if this is a circular chromosome, we come back around to where we started at, and DNA pol 3 will run into the primer from the other end. Then what's the next enzyme that plays a role?
The polymerase 1?
Yeah, DNA pol 1. What does DNA polymerase 1 do?
Replaces the primer with DNA?
Right. So DNA pol 1 is able not just to add new nucleotides, but it can cut them out too. So it's going to find the primer and cut out all of those RNA nucleotides in the primer. Then it's going to find the end of that newly replicated daughter strand that came around from the back to meet up the back of the primer, and it's going to start adding nucleotides there until the whole primer is replaced with DNA nucleotides. And then you have your last enzyme, the ligase, which does what?
Joins the DNA of the replaced primer and the rest of the strand.
Right. Yeah, so your DNA polymerase 1 or 3, they can add new nucleotides to an existing strand, but they cannot join two strands together. So once this primer is replaced with DNA, you would need to join it to like the rest of the daughter strand, and the DNA polymerase can't do that because that's already an intact strand. So you have ligase come in and just make a phosphodiester bond between the end of the newly replaced primer and the rest of your daughter strand. Now you finally have a complete daughter strand. Okay, and that's basically how things go for the leading strand. So what's the difference between the leading strand and the lagging strand?
It's the leading runs the 5' to 3', and then lagging is the opposite, 3 to 5.
Right. So with the leading strand, that's one of the template strands of your original DNA, and it's oriented 5' to 3'. So your helicase would be moving in that same direction, 5' to 3' along the double helix, so that you always are going to have like a new 3' end opening up, or new spaces where you can add nucleotides to the 3' end of the daughter strand that you're building. So your polymerase, your DNA polymerase 3 on the leading strand is able to just follow along behind the helicase. The helicase opens up a new stretch of the double helix, and the polymerase will just follow in after it, adding new nucleotides to the daughter strand is making to the 3' end, which is always pointing towards the helicase. Nice and simple-ish. Then for the other strand, since the DNA double helix is anti-parallel, the other strand is running the opposite direction. It's running from 3' to 5', which means that it would actually be the 5' end that's pointing towards the helicase. So the helicase is going along the DNA, opening the double helix, but it's exposing more, I guess, more of that template strand on the 5' end of the daughter strand that's being made. And DNA polymerase can't add to the 5' end. If it was going to follow the helicase, it would need to be able to add to the 5' end, but it can't do that. It can only add to the 3' end, which is pointing away from the helicase. So that means that on the lagging strand, DNA polymerase has to go backwards to make the daughter strand. So it can only go back so far, of course, because you're going to run into like the double helix wherever helicase had not opened it yet. So it's going to go as far back as it can, and then it's going to do what?
Separate?
Yeah, it's going to detach from where it's at. And where is it going to reattach at?
Is it after the primase?
It's going to, well, yeah, actually. First, you would have to have primase come into this new location and make the primer, and it's going to come in and start adding to that primer. But this is basically all going to be like, it's going to all hop back closer to what the helicase is now. So if you're starting here, your DNA polymerase on the lagging strand has to move this way. The helicase, meantime, is moving this way. So they're both moving. Your DNA polymerase will have made like this much of a daughter strand, and then it's going to run into the double helix again and have to stop. So it's going to detach and hop back to where the helicase is. Technically, you're going to have primase there first, making a primer, and so the DNA polymerase will attach to that primer and then add in the rest of the daughter strand until it runs into the part that it made before. So it's going to be making this daughter strand in little chunks like that. Every chunk is called an Okazaki fragment. for every single chunk, you're going to have primase making a primer, then DNA polymerase 3 adding to that primer, making the daughter strand, and then DNA polymerase 1 is going to come back in and replace every primer with DNA. And then every primer is also going to have a ligase come in and join it to the next fragment of the daughter strand until eventually you have constructed a complete one. Cool. So what's the reason, in terms of the DNA polymerase limitations, what's the reason why you have the leading and lagging strands? Why can't they both be the leading strand?
Is it because like on each strand it has opposite nucleotides?
Right. On each strand, well, I guess they're antiparallel, so the nucleotides will be oriented in opposite directions. And then DNA polymerase can only add to one end. It can't add to both ends, only one. So that means that it has to go whichever direction that strand is pointing. One is pointing towards the helicase, the other points away from the helicase. Okay. Then we have transcription. So what is transcription?
Making
The
RNA
process of
from
making
DNA.
RNA from DNA.
Right, making RNA from DNA. Usually would be making mRNA, but actually any RNA that's made from DNA would be made in transcription. So what's a promoter?
Where it starts from?
Right, that's where transcription is going to begin. Where would you find promoters in the chromosome?
In nucleotide sequences in front of each gene type?
Yeah. Right, so in front of every gene, you're going to have a promoter, or in front of every operon, you'd have a promoter. So for genes that are together in an operon, each one doesn't have its own promoter, just the whole operon has a promoter. And that's where transcription is going to begin. So you're going to be able to transcribe whichever gene is like next to that promoter, or whichever operon is next to the promoter. How is the RNA polymerase going to attach to the promoter in bacteria?
Is it the sigma factor?
Yeah, sigma factor. What is sigma factor?
It's an RNA subunit.
Yeah, right. That's a subunit of the RNA polymerase. So the sigma factor will attach to the promoter. Then since that's a subunit of the polymerase, it just brings the whole polymerase with it. So that's for bacteria. How does RNA polymerase attach to the promoter in Archaea?
Transcription factor?
Yeah, transcription factors, exactly. What's a transcription factor?
In the open region?
They're going to be binding to part of the promoter, to certain regions of the promoter.
I guess proteins that bind.
Yeah, right. They're just proteins. So they're not a subunit of the polymerase. They're like just different independent proteins that are going to be able to bind to the promoter. And each one will have kind of a different part of promoters that it binds to. So each transcription factor wouldn't necessarily be able to bind to every single promoter. It depends on if that promoter has the specific sequence that it's able to bind. Then how does RNA polymerase attach to the promoter in eukaryotes?
I think in eukaryotes and Archaea, they both use the transcription factors.
Yeah, exactly. So in eukaryotes, again, you have transcription factors that come in and bind the promoter, and then RNA polymerase is able to attach to those transcription factors. What is the difference in how that happens in eukaryotes versus Archaea?
But in eukaryote and Archaea, they bind the promoter first, and then they bind the RNA polymerase.
Yeah, right. So in both of those groups, the transcription factors bind first to the promoter, and then second, RNA polymerase will come and attach to the transcription factors. There's actually a difference in the transcription factors between Archaea and eukaryotes, though.
Oh. Is it Archaea? They have several, and then eukaryotes have many?
Yeah, exactly. So in
Okay.
Archaea, you just there's just a few different ones in Archaea, but in eukaryotes, there's just like many different transcription factors. So many different ones. Then in Archaea, since there's only a few, you might need just maybe one to bind or a couple to bind, then the RNA polymerase can attach. In eukaryotes, you're going to need like a number, like several different transcription factors are going to have to be attached to the promoter all at the same time before RNA polymerase can bind to that promoter. So that allows us to have a lot more, I guess, sophistication in our control of which genes we're transcribing, which maybe a microbial eukaryote doesn't have a use for, but for eukaryotes like you and me, we really need to have tight control over transcription and which genes are being transcribed at any given time, because we have different cell types in our bodies, and all those different types all have the exact same genes. And we have to make sure that they're not transcribing the genes that are for a different cell type. So that's the reason why we've got so many transcription factors, and you need a bunch of them to bind to the promoter all at the same time before you can transcribe the gene. Actually, you guys all know examples of some transcription factors because you've heard of estrogen and testosterone. So those are actually both transcription factors. Yeah. Oh boy. Changing the formatting of my study guide. Okay. What's the difference between the template strand and the coding strand?
The template this has the yeah.
Sorry.
For the template strand, instead of the T, it's the U?
That's going to be the coding strand, actually.
Okay.
What are you using the template strand for?
Is it to bind the RNA?
Yeah, right.
Okay.
So when the RNA polymerase is building the new strand of RNA, each nucleotide, each RNA nucleotide is going to be binding to the template strand or base pairing with the template strand. And then when it base pairs correctly, RNA polymerase will add it to the new strand of RNA that it's building. So the template strand is containing the instructions basically for building that new RNA strand, and the new RNA strand you're building is going to be base paired to that template strand, at least for a little bit. Then the coding strand is the other strand of DNA that you are not using for transcription. Okay, so in replication, you're going to replicate both strands of DNA, and both of them will be a template strand for its own daughter strand. Then in transcription, you only use one strand as the template strand. The other strand is called the coding strand or the complementary strand. And it's called that because it's going to, well, it's called complementary strand because it can base pair with the template strand. And it's called the coding strand because it's going to have the same information as the RNA has that you're making. The only difference is that it's going to have T's and the RNA you're making will have U's. So the sequence is going to be the same. Since the coding strand needs to be able to base pair with the template strand, and the new RNA strand that you're building needs to base pair with the template strand, both of them are going to have the same sequence if they're able to base pair with the same template strand, except that, of course, you don't have T's and RNA, you have U's. Okay, so I guess I basically kind of said it already, but what's the role of the RNA polymerase in transcription?
It adds the nucleotides to the RNA strand.
Right. Yeah, so that's going to be attaching to the promoter and then building an RNA strand that is complementary to the template strand. So it's going to be walking down the gene from the promoter, and it's just going to be having RNA nucleotides come in, base pair with the template strand. If the base pairing is correct, RNA polymerase is going to add that new RNA nucleotide to the RNA strand that it's building by catalyzing phosphodiester bond formation. So it's building your new strand. So we've got like the first parts of that basic process. We've got that transcription is beginning with either sigma factor or transcription factors attaching to the promoter, then RNA polymerase attaching to those things, then RNA polymerase builds your daughter strand of RNA. And then basically, I guess, it's all going to finish when what happens? Or I guess, how is it going to finish?
I'm not sure.
So it's going to finish when you have some type of termination sequence that's transcribed into the new RNA strand that you're building. So there's different ways that can happen, and we're not going to worry about the different ways, but there's some type of termination sequence that the RNA polymerase is going to get to and transcribe, and then once it's transcribed that, it's just going to let go. Everything is going to separate from everything else. Transcription will be over, and you'll have your new piece of RNA. Okay, then RNA processing. So what type of organisms do RNA processing?
Eukaryote?
Yep, eukaryotes. So actually, prokaryotes will also process RNA, but they just don't do it like as much. Have very limited processing of RNA that they do. Eukaryotes do pretty much a lot of RNA processing. So we basically focus on RNA processing in eukaryotes, and we ignore it. You know, not everybody. I guess there's scientists who study this, but for us, we ignore it in the prokaryotes. So part of the RNA processing, or I guess what are some things that are done in RNA processing?
Cutting.
You're going to cut the new RNA strand? Yeah, that's part of it. What happens after you make a cut? After you cut something out, what happens next?
After cutting it out, like exons start to flank together.
Right. And what do you call the piece that got cut out?
An intron?
Yeah, an intron. Yeah. So every bit of mRNA that's just been made is going to have regions that are exons and then regions that are introns. Part of the RNA processing is RNA splicing. In RNA splicing, you're going to cut out the introns and then join the exons together. So if you had two exons flanking an intron, so they're on either side of that intron, once the intron is cut out, they're just going to be brought together and joined together. So generally, you're going to be cutting out all of the introns and joining together all of the exons. And the exons are for keeping, the introns are for cutting out. So that's RNA splicing. That's a big part of RNA processing. What's another thing that happens in RNA processing?
Addition of the 5' cup and alpha A.
Yeah, right. You're adding the 5' cap and the poly A tail. So which end of the new RNA strand does the cap go on? I guess the name kind of tells you.
The 5'.
Yeah, the 5' end. And which end is the poly A tail going to go on?
The 3'.
Yeah, the 3' end, exactly. What is the 5' cap used for?
Translation?
Yeah, it has a role in translation. Specifically, it's going to allow the ribosome to attach to the mRNA. It's actually going to be grabbing onto the cap, to the 5' cap. And then what's the job of the poly A tail?
Protects the mRNA from degradation.
Yeah, protects the mRNA from being degraded. So for eukaryotes, they don't like to have just kind of strange pieces of RNA floating around in the cytoplasm. If they find some RNA just floating there, they are going to degrade it. So that would include their own mRNA, unless it has the tail, the poly A tail. If they see a piece of RNA that has the poly A tail on it, they're not going to degrade that. And it's basically a defense mechanism. In general, if there's some strange RNA floating around in your cytoplasm and you're a eukaryote, probably that means that RNA belongs to a virus, and you're being infected right now with the virus. So it's good to degrade that RNA and get rid of it and do not turn it into a viral protein by translating it. But then you need a way to mark that your own RNA is like good and safe, so they mark it by putting the poly A tail, which is called the poly A tail just because it's a lot of A's that are added. So at the end, at the 3' end of the mRNA, you're just going to have a polymerase come in and just add a bunch of A's to the end and make the poly A tail. Okay, cool. Once your RNA is processed, then you can finally translate it into a protein. So what is translation? I guess I kind of maybe said it.
Oh, wait, did you ask what was translation?
Yeah.
Oh, isn't the process of making mRNA, or when mRNA divides?
So that's actually transcription. When you're making, anytime that you're making RNA, but usually it would be mRNA, that's transcription. And then when you use the mRNA, that's translation. And what would you be using it for?
To make proteins?
Yeah, to make protein, right? So in translation, you're using mRNA to make a protein. I guess technically speaking, we should say that in translation, you're using mRNA to make a polypeptide because we can't call it a protein until it folds up.
Okay.
But, you know, we'll just go with protein. It's kind of a technicality. Okay, so what's the role of the mRNA in translation?
Specifies what amino acid goes where.
Yeah, right. So the mRNA is containing the information that's from that gene that's telling the ribosome which amino acid needs to go in what spot, or what should the sequence of amino acids be for this new protein. Then what's the role of the ribosome?
To like copy?
In a way, it's like to copy the information from that mRNA into a protein. Yeah. But of course, you're not building something out of nucleotides when you're making the protein. You're building something out of amino acids. So it's like copying, but it's also not like copying because you're using a different building block to build a different type of macromolecule. So the type of macromolecule is changing completely, but the information is remaining the same. That is actually being copied. Within the ribosome, you have rRNA, or ribosomal RNA. What is the job of the rRNA?
To catalyze the peptide bond formation?
Right. So the rRNA is actually the part of the whole ribosome complex that is catalyzing the formation of peptide bonds and linking amino acids together. So the ribosome is kind of the factory where this whole process is occurring. And it's doing the process as well, but specifically it's the rRNA part of the ribosome that is actually linking those amino acids together. Then what's the role of tRNA?
Brings the amino acids to the ribosome.
Yeah, brings the right amino acids into the ribosome. So it's making sure that the correct amino acid comes in, whichever amino acid was specified by the mRNA. Then what's the role of release factor?
It separates the polymerase from tRNA and making the ribosome to dissociate.
Yeah, right. Release factor is going to be coming in at the very end of translation, and it basically makes everything fall apart. So the ribosome is going to come apart. The mRNA is going to come apart from the ribosome. The tRNAs will leave, and the new polypeptide that you made is going to leave as well. So everything will just let go of everything else, and translation will be over once release factor comes in. So we've got 3 phases for translation: initiation, elongation, and termination. What's basically happening in initiation?
In initiation, the ribosomes assemble on the mRNA and then ensures the correct location.
Yeah, exactly.
Or they'll be read wrong.
Yeah. So in initiation, everything is basically getting set up. You have your ribosome attaching to the mRNA. First the small subunit will attach, then the large subunit will attach. You also have your first tRNA coming in, and you're making sure that the mRNA is positioned correctly in the ribosome so that the ribosome will start translating at the right spot. So how is it that the ribosome is going to attach to the mRNA in a prokaryote?
Binding site?
Yeah, the ribosome binding site, or the RBS, which in 1306, when we talk about this, we call it the Shine-Dalgarno sequence, which it's named after the guy who discovered it. But our current textbook is going with the ribosomal binding site, or RBS. So I'm just going to use both terms on the test. So, whichever one is easier to remember, you can remember. Right, so for prokaryotes, the ribosome, the small subunit of the ribosome, is going to find the RBS. It's going to be close to the front of the mRNA is where you have your RBS. It's going to find that RBS and attach to it, and then that makes sure that the rest of the ribosome assembles on the mRNA in the correct spot. Let's see. Then in eukaryotes, how is it that the ribosome attaches to the mRNA?
The 5' cap?
The 5' cap, yeah, exactly. So for eukaryotes, the ribosome small subunit will grab that 5' cap, and that makes sure that everything will assemble in the correct position. When everything is assembled in the correct position, the first codon that the ribosome will read is going to be the start codon, which is why we call it the start codon, because it's the first. Then you're going to have also your initiator tRNA come into the ribosome and pair with the start codon. Which part of a tRNA molecule would pair with a codon?
The 4-methylmethionine tRNA?
That is the initiator tRNA for prokaryotes. So they'll have a tRNA that has a special amino acid attached to it, which is the formal methionine. So methionine is a normal amino acid, but the formal methionine is not normal, it's modified. only the initiator tRNA is the one that has that formal methionine. So only the formal methionine tRNA is going to pair with the start codon in prokaryotes. In eukaryotes, it'll just be a tRNA that has regular methionine on it. But it's not the methionine end that's actually going to pair with the codon or the formal methionine end. It's going to be something at the other end of the tRNA.
The exit?
Let's see. One of the sites in the ribosome where tRNAs can be present in the ribosome is the exit site.
Would it be the A site?
The A site is going to be the site where they enter. So these are places in the ribosome where you can put a tRNA. Actually, in initiation, we're going to have the tRNA in the P site. The start codon is going to be lined up with that P site. So the initiator tRNA comes into the P site and it pairs with the codon. And it's important that this all happens in the P site. That way, everything is lined up correct, and we know that we're starting in the right spot. But it's actually going to be a different part of the tRNA that lets it pair with that start codon. So every tRNA is going to have kind of two ends on it. One end has the amino acid that's attached to it, then the other end has something else, something that can pair with the codon.
Polypeptide chain?
See, that's going to be what you're building ultimately out of this. The name is actually similar to codon, but a bit different. It starts with an A.
The acceptron?
Not the acceptor site. But it's a different A word. Starts with an A and ends with codon.
Anticodon?
Anticodon. There we go. Cool. So every tRNA has on one end an anticodon, and on the other end the amino acid that's attached to it. The anticodon is 3 base pairs. Well, no, not 3 base pairs. 3 bases, 3 nucleotides able to base pair with the codon. So the codon on the mRNA has 3 bases in it. The anticodon has 3 as well, and it's going to be the complementary 3. So if your codon is AUG, which is actually the start codon, then the anticodon has to be UAC. There we go. To pair with the codon. So your initiator tRNA comes in. It's going to enter the P site on the ribosome. It's going to have its anticodon base pair with the codon, with the start codon, and it's going to have its proper amino acid attached. So now everything is set up right. You have your initiator tRNA. It's correctly attached to the start codon. The start codon and that tRNA are both there in the P site. Everything is good to go now. So now we're going to move to the elongation phase, where basically you're building that polypeptide chain. So what's the first thing that's going to happen in the elongation phase?
The polypeptide is built?
Yeah, so that's the ultimate thing that you're doing, and it's going to go, I guess, in a little cycle. Every time the cycle runs, you're going to add 1 more amino acid to that polypeptide that you're building until you get to the end of the mRNA. When you are going to be adding an amino acid to that polypeptide that you're making, what's the first thing that has to happen to add a new amino acid?
Sorry, can you repeat the question?
Oh, okay. Forgot my own question. When you're going to add a new amino acid to this polypeptide that you're building, what's the first thing that needs to happen? What's the first step in adding a new amino acid?
A new tRNA enters.
You're going to have a new tRNA come into the ribosome. Yeah, exactly. Which site is it going to enter at?
The A site.
A site.
The A site. Yeah, exactly. So you have a new tRNA that comes into the A site, and it's going to have its anticodon base pair with the codon that's on the A site. So now your new tRNA is in the A site. What's going to happen next?
The rRNA will catalyze the peptide bond formation.
Yeah, exactly. So your rRNA in the ribosome is going to catalyze the formation of a peptide bond between the amino acid that's on this new tRNA in the A site and the amino acid that's on the tRNA in the P site. So now those two things are linked by a peptide bond, and you're going to have those amino acids let go of one of those tRNAs. Which tRNA do they let go of?
Translate.
Let's see. It's going to let go of one of the tRNAs. Will it be the P site tRNA or the A site tRNA?
Oh, I think the P site.
The P
The
site,
A
yeah.
site? Oh, P
It's
site?
going to let go. Yeah, it's going to let go of the P site tRNA, which means that everything now, those 2 amino acids with the peptide bond, and whatever else you had in your polypeptide chain already, all of that is now going to be attached to the A site tRNA. And then I think now if you started to say what happens next.
The trisolate move?
The translocation, yeah. So the ribosome is going to translocate or walk down the mRNA. It's going to move 3 bases down the mRNA, or 3 bases, which is the same as 1 codon. So that means that your tRNAs that are base paired with their codons are going to be shifting inside of the ribosome. So your P site tRNA is now going to be in which site?
E site?
In the E site, yeah. And what happens in the E site?
The tRNA exits.
Yeah, that's the exit site. So the tRNA that's there is just going to leave. Then your A site tRNA, which site is that one going to end up in?
The P site.
The P site, exactly. So now your polypeptide chain is attached to the tRNA that's in the P site. And your E site tRNA is gone, and you've basically completed this cycle, and you're ready to start again. So next you'd have a new tRNA come into the A site. Our RNA will catalyze peptide bond formation between this new amino acid and the existing polypeptide chain. That chain is going to be transferred to the A tRNA, the A site tRNA. Then the ribosome will translocate, walking 1 codon down the mRNA. Your tRNAs will shift, so the A site tRNA is going to go to the P site, and the P site tRNA goes to the E site and leaves. So this is a cycle that's going on in elongation, and it just happens over and over until eventually you get to the end of the mRNA. And then you're going to have termination, in which the whole thing stops. So which codon is going to cause termination to occur?
The stop codon?
Yeah, the stop codon, exactly. So when your ribosome translocates at the end of your mRNA so that the stop codon comes into the P site, or I should say actually the A site, then the whole process is going to stop. You're going to have a protein that comes in. Which protein is going to come in to the A site when the stop codon is there?
You said which protein comes in?
Yeah, it's not a tRNA this time. It's going to be a protein that kind of looks like a tRNA.
Is it release factor?
Yep, exactly. So when you have your stop codon enter the A site in your ribosome, release factor is going to come in. There is no tRNA that can pair with a stop codon. So instead, release factor is going to come into the A site, and it looks similar to a tRNA, but it is not a tRNA. Once the release factor is in there, everything is going to let go of everything else, and translation will be over. So that's termination. Okay. Then the last thing we have under translation is code degeneracy. So what is code degeneracy?
Degeneracy. multiple codes can specify the same amino acid?
Right, yeah. So every amino acid, let's see, can have multiple codons that mean that same amino acid, or multiple codons can mean the same amino acid. So for every individual codon, it's always going to mean that amino acid that it means. But you can also have another codon that also means that same amino acid, and that's the code degeneracy. Why is that important?
Reduces the impact of mutations.
Right. It's reducing the impact of mutations. So actually you don't need to know like the wobble pairing for the test, but the reason why you have code degeneracy is that when the tRNA anticodon pairs with the codon, the last base there in that anticodon actually does not pair normally. The anticodon is kind of curved, and the last base is curving up. So it's at an angle. It's not pairing straight on with the codon. It's pairing at an angle, and that's wobble pairing. So since it's at an angle, it's not actually able to base pair really like correctly, which means that it kind of doesn't matter if it would pair correctly or not. So, if you had a G here and your last position in the codon, you should have a C in your anticodon to pair with it. Since it's at an angle, it actually doesn't have to be a C. It could be an A, maybe it could be a U. Something else would also be able to pair with that G, which means that any codon that starts with like the correct two bases is going to end up meaning that anticodon, or meaning a range of anticodons, that is. No, wait, meaning the same anticodon, sorry. One given anticodon will be able to pair with multiple different codons if they differ only in that last base. The first two bases have to be the same. The last base can be different, and the anticodon will still be able to pair. So if you had a mutation that occurs at that last base in the codon, usually it's actually going to not make a difference. Since that wasn't going to be a correct base pair anyways, you can still have the same tRNA pair with that codon, even though it's different now. If you have a mutation that's to the first two bases, that is a problem. That's going to mean that a different anticodon will bind, and you're going to get a different amino acid in that protein. If it's just the last base that is different, or the last base that's mutated, you can still have the same anticodon bind, and the same amino acid will show up in that protein. So the benefit of the code degeneracy, or how it reduces the impact of mutations, is that if you have a mutation that's occurring at the last base in a codon, you're probably going to end up with the same amino acid in the protein. If you have a mutation that's in the first two bases of that codon, you're going to get a different amino acid, which might affect the function of the protein. If you have just the mutation at the last base, you'll probably get the same amino acid, which means that there's not going to be any effect of that mutation for the organism. Its protein will still look the same, function the same, and be the same. Okay, so that's the last of translation. Technically, what we would make from translation is not the full protein, it's the polypeptide, because it still has to fold and gain its functionality before we can technically call it a protein. So after translation, that polypeptide is going to go through protein processing. One of the main things that happens in protein processing is that the protein folds up to become a fully functional protein. Which molecule is going to help the protein fold?
It's chaperones?
You're going to have a chaperone help the protein fold. How does the chaperone do that?
It can refold the denatured proteins?
Yeah, it can. Some chaperones can. So if your protein denatures, which means it loses its shape and therefore its function, sometimes you can save that using a chaperone to refold it. How would that happen? How is a chaperone going to be refolding a protein or making it fold the first time?
By assembling the multiple polypeptide subunits and associating them with the cofactors with the enzymes.
So you do have some chaperones that can do that, like help your subunits come together the right way, help your cofactors come together with their enzymes the right way, so that everything comes together nice and hunky-dory.
Does it use heat?
Sorry?
Does it use heat?
Some chaperones can insulate the cell from the effects of heat by refolding proteins that were denatured due to heat. So you have your heat shock proteins, and those are chaperones that they're not using heat, but if the cell is exposed to like high heat, the heat shock proteins will be able to refold some of the denatured proteins. So it's helping the cell be protected from high heat. If you have a chaperone that's going to be folding up, like let's say a polypeptide that's going to be the subunit for a protein, how is it going to do that? So is it actually going to like take this polypeptide and like physically fold it up, or is it going to do something else?
It's gonna isolate it.
It's going to isolate it. Yeah. So that's one thing it can do. It can isolate and well, I guess it can isolate the polypeptide from the environment, but also it can isolate like specific parts of the polypeptide from other parts of the polypeptide. And what's going to be the benefit of that?
It's gonna allow it to take its time.
It's going to, well, I guess in general, chaperones are going to make this process go more quickly. So for any protein, or any polypeptide, you're going to have a lot of different ways that it would be able to fold up. It doesn't necessarily have to happen just one way. And it's going to generally take a long time for that to happen by itself. So the chaperone is going to be making that happen more quickly, and it's going to be making sure that incorrect foldings do not occur. So one of the ways that it could do that would be isolating one part of the polypeptide from another part of the polypeptide. If those two parts cannot come together, then they're not going to interact and fold wrong with each other. Then it's going to be only the parts that need to fold with each other would be next to each other and would be able to fold. So the chaperone is not actually like causing the protein to fold correctly, but it's making sure that the protein does not fold incorrectly. So if it sees that there's kind of a wrong folding occurring, it's going to stop that from happening or prevent it from happening in the first place. And then once all of the wrong patterns are blocked, only the right pattern remains. And also, it's going to just be making this process go much more quickly. So that's your chaperones, preventing wrong foldings and speeding up the process to fold your polypeptides into a functional protein. And then also, if your protein has subunits, making sure the subunits come together correct, making sure cofactors come together correct, and refolding denatured proteins. Okay, so now you've got your folded protein, and it's finally technically a protein because it's folded and it has a function. So for prokaryotes, most of those proteins are just going to be, they're done now. They're going to chill out in the cytosol and do their job. But sometimes you might need to send a protein to a specific location. And then in eukaryotes with their organelles, there's a lot of different places where they need to send a protein. So once the protein is done, it needs to go to the correct destination for it, wherever it needs to do its job at. So for a prokaryote, that's going to be outside the cell. For a eukaryote, that might be outside the cell, or that might be a different specific organelle within the cell. Other than that, if the protein doesn't have a specific special destination, then it'll just kind of be released and float away into the cytosol. So if you're a prokaryote and you need to send a protein outside of the cell for it to have its proper function, how are you going to make sure that it gets to the outside of the cell?
Use a specific or special amino acid.
Sorry, what?
It uses special amino acids.
Excuse me. Yes. Yeah, so you're going to have a special sequence of amino acids at the front of any prokaryotic protein that needs to go outside the cell. What do we call that special sequence of amino acids?
A single sequence?
A signal sequence, exactly. Yeah, so for prokaryotes, either your protein is just going to stay in the cytosol where it's at, or it's going to go outside the cell. Any protein that has the signal sequence, that special sequence of amino acids at the front end of it, if it has that signal sequence, it's going to be targeted for going outside the cell. It's going to end up interacting with the machinery that would pump it out of the cell. Then for eukaryotes, they're kind of more complicated. They have a lot of different places where they might want to send a protein. So sometimes they're going to use a little tag that's made out of amino acids, kind of similar to the signal sequence, just some specific sequence of amino acids near the front of the protein that's going to send it to a specific place within the cell or outside the cell. But they can also target proteins in other ways. So instead of using an amino acid tag, they could also use a tag made of what types of things? What type of molecule is added to proteins when they go to the Golgi apparatus?
When they go to the Golgi apparatus, a protein?
Yeah. Well, I guess it's going to be not a protein. The proteins are going to go to the Golgi apparatus, and then something else would be added to them there, not a protein, but a different type of biological molecule.
Would it be an enzyme?
An enzyme is going to be a type of protein. So it's actually, it would be an enzyme that's doing this. You're going to have enzymes in the Golgi apparatus grabbing these new proteins coming through, and then those enzymes are going to be attaching something else to them.
Ribosomes?
Let's see, the ribosome is going to be left behind already. So this protein would have probably come through a ribosome that's studied on the surface of the rough endoplasmic reticulum. It would have been made by that ribosome, extruded into the rough endoplasmic reticulum. Then the RAR would have packaged it up into a vesicle and sent it to the Golgi apparatus to be sorted for its correct destination. And in the Golgi apparatus, you're going to have special enzymes that add different things to the protein. So it's going to be one of our types of macromolecules, actually. So we had, what are the types of macromolecules?
Is it a lipid?
Ah, that's a good guess, but it's not a lipid. Actually, sometimes it would be a lipid, to be honest, but usually not a lipid.
A carbohydrate?
A carbohydrate, yeah, exactly. So in the Golgi apparatus, you're going to have different sugars being added to these proteins coming through. Each protein that goes through the Golgi apparatus is going to get a tag added to it that's made of like a different combination of sugars in like a specific pattern. And then that tag is going to send it to where it needs to go. So for eukaryotes, for prokaryotes, if they need to go outside the cell, they'll have the signal sequence, which is a sequence of amino acids at the front of the protein. For eukaryotes, proteins that need to go to specific destinations are going to have some type of tag. Sometimes that tag will be a sequence of amino acids at the front end. Sometimes it's going to be a series of carbohydrates or a series of sugars that is added to that protein, tagging it to the correct destination. Okay, and that is chapter 4. Okay, so I guess we're a little bit over time. Are there any kind of last minute questions you guys had?
I don't think I have any. I just needed the explanation for chapter 4, which is what we just did.
Right, for sure. Okay, cool. If you guys think of anything, you can always email me. And cool, okay. Well, thank you guys for coming.
Thank you.
Thank you.
And I hope it was helpful, and I hope you guys have a good night.
You too,
Thank you, you too.
Bye.